The overall goal of the following experiment is to demonstrate a protein-receptor interaction using surface plasmon resonance. This is achieved by injection of the nickel ions over the nitrilotriacetic acid, or NTA, matrix. Next, one of the proteins, termed the ligand, is immobilized onto the sensor chip. Following a blank injection, the second protein, termed the analyte, is injected over the immobilized ligand in order to detect an interaction between the two proteins. The results show that the two proteins interact transiently. The analyte associates and dissociates from the ligand. The affinity of the interaction can be calculated based on the measured association and dissociation rates of the two proteins.
The main advantage of this technique over existing methods like pull-down or immunoprecipitation is that it can identify transient or low affinity interactions that are often of great biological consequences.
This method can help answer key questions in biochemistry, such as whether the protein of interest binds to another protein or another small molecule. The nature of the interaction can be determined and the affinity can be measured.
To prepare the sample, remove all aggregates from the purified protein as described in the text protocol. Prepare the running buffer and filter all buffers and protein samples using 0.22 micron filters. Ensure that the filters are protein compatible in advance. Before connecting the running buffer bottle to the pump inlet, keep aside 10 milliliters of the running buffer in a separate tube. Then, dilute the samples against the running buffer to avoid buffer mismatches.
As a rule of thumb,